I had to wait 10 seconds for the online participants to be loaded, but yeah, so thanks very much for coming today. I'm really happy to welcome David Brainard, who's visiting from the University of Pennsylvania. Also welcome to all the people who are online, who are attending this remotely as part of the Sussex Vision series with Worldwide Neuro. So David has done research in many areas of colour vision science and vision science more broadly. You might have come across his work directly in various topics, but in particular, David has co-authored some of the most widely used toolboxes in vision science. So the Psych Toolbox, which is a MATLAB toolbox for running psychophysics and doing data analysis and colour imagery and all kinds of things, and also the ISET Bio Toolbox, which is a toolbox that aims to model retinal responses to light. So modeling the optical image and all the optical distortions that are introduced as light travels through the eye, and then retinal signals in response to that light. So this is a really useful toolbox. Be a good thing to look into if you haven't looked at that already. So David today is going to be talking about his work on geometry of color space. Thanks, David.
Thanks, Jenny, and thanks everyone for coming and to listen to me talk about some of our current work. This is a project that's about 3 years old, and before I start, I just really want to highlight Phil Guan, who is our scientific collaborator at Meta, Alex Williams. This project actually started over coffee. I was on sabbatical at the Flatiron Institute for Computational Neuroscience, where Alex said, I have some methods that might be useful for characterizing color. Do you have any interest in talking about that? And the answer was yes. And then particularly Fangfang Hong, who's a postdoc, who's really integrated together Alex's ideas with our knowledge of color and driven this project forward. So let's see, can I advance my slides? Oh, there we go. Great. This is a color center at the university with lots of experts, but I do want to just start out and get us all on the same page about some basic facts about color before I go on to new things, just to make sure for those of you who may not specialize in color, we've sort of got the basics under our belts. So human vision is trichromatic. There are 3 classes of cones that code spectra of light. They have distinct spectral sensitivities, the LMS cones. Any light spectrum is coded as far as the rest of the visual system goes, the rest of the brain, by the excitations that this light causes in those 3 cone classes. And that's what makes us trichromatic. It enables color reproduction by mixing red, green, and blue primaries. It also means that we can represent color stimuli in a greatly reduced fashion relative to the physical signal as a point in a 3-dimensional color space. And this just illustrates one such space where the axes are the excitations of the 3 classes of cones. So this is a Cartesian coordinate system, and a point here corresponds to some triplet of the excitations. And just with that idea, something that many of you may know is that if 2 different lights, 2 different spectra, say a natural occurring spectrum that might encounter outdoors and a light from, say, a computer display that was reproducing that, have different physical spectra, but happen to have the feature that they produce the same excitations in all 3 cone classes, then we call them metameric. They are indistinguishable to a normal human observer because the information available is the same. And it's really this kind of identity that's driving the technical underpinnings of color reproduction. And those 2 stimuli will point to the same point in color space. So one thing we can say is if we're thinking about the geometry or geometrical representations of color, this trichromacy puts some important constraints on it. Spectra that are different in the world, yet produce the same excitations in the cones, go to the same point, and so we can map stimuli into a space that represents those excitations. So it puts constraints on it, but it doesn't fully determine what I'm going to introduce in a second, other geometrical features of our representation of color, because we could stretch these axes or rotate them or really transform in any invertible way, and a single point in the original cone excitation representation would still be a single point in a transformed representation. And so if that transformation is invertible, it also would capture this aspect of human color vision. So this geometry is not the only one that we could have that represents color. And in particular, if we had now considered 2 different stimuli that produce different sets of excitations in the 3 classes of cones, those would plot to different points in this cone space and in other cone spaces that were an invertible transformation away. And a natural question that I think is what I mean by when I talk about the geometry of color space is that if we have stimuli plotting to two different points, how perceptually different are they? Can we tell them apart, or are they indistinguishable? And if we can tell them apart, how similar or dissimilar do they look? That idea that we might be able to find a geometric representation of color stimuli based initially on the trichromacy on the cone representations, but that captures other aspects of color vision is really a long-standing and very fundamental question in color vision, and actually in perceptual science more generally in other domains where we'd like geometric representations of the perception. I'll just comment, and there's been a lot of work on this question over the past even 150, almost 200 years now. A lot happens to the cone signals along the visual pathways before their effect arises at our percepts and allows us to make judgments. And so there might be a little optimistic to think that just the kind of straight line Euclidean distance between 2 points in this particular space would actually capture other important aspects of perception. So, but that's really the question I want to come back to, and it's a question that, as I say, this kind of new research program for me over the past several years that we've been having a lot of fun with it. We think it's important, but that's the basic question. And I think it's worth just saying for a minute, why do we care about the answer to this question? Why might you care? And I think there's a couple broad reasons, or three broad reasons, that we certainly try to keep in mind as we work on this. First of all, to the extent that we could develop models that predicted not just when two colors were identical, but how different did they look, or were they discriminable when they didn't produce the same cone excitations, that's going to put constraints eventually on models of what happens to the cone signals as they travel along the visual pathway. That is to say, if we have a mechanistic model of color vision, it ought to be constrained by and predict these kinds of behavioral results that I'm going to talk about. So that's one reason. I'll say just a little bit about how we're using data we're collecting to that question today. Secondly, and I know there are people here who really are interested in color cognition and cognition kind of at a level that builds on perception more generally. And so questions of memory, attention, categorization are examples of processes that there's a great interest in understanding, core psychological processes. And often when one studies those kinds of things, what you would like is to be able to have a set of stimuli, and people often use color with the hope that this could be done, where you could, in some principal way, say that the perceptual distance between stimuli had been equated so that effects you wouldn't misattribute to categorization or attention or memory, effects that could just be explained by a non-homogeneity of the distances between the perceptual stimuli themselves. So you really want a theory, and particularly a metric theory, that lets you add distances in a psychologically meaningful way that would support work of this sort. And I think color is used in some ways because we know more about it and it's multidimensional than other systems, but we don't, I think, yet know enough about it. And finally, and we were talking at lunch today about relationships between industry and basic science and applied research. There's a lot of desire when you build a device or compress an image or transmit an image. You might want to not have to transmit or render or capture with a camera information that people can't see, that isn't relevant to the actual percept of color. And if we understood exactly those kinds of things, we had theories of that could be expressed geometrically or really in any other way, then we could provide, I think, better advice or check whether the advice we're giving to people who are trying to do that kind of engineering based on color, that could be helpful. So those are three broad reasons that have motivated our interest in this question beyond, and sometimes I am a kind of curiosity-driven scientist at heart, or at least a large part of me is. And despite these other interesting applications, I just find this a really interesting question of the geometry of color. So one thing in perceptual science that we actually have learned how to do, I think, very well and repeatably and reliably and rigorously is to measure what we call perceptual thresholds. How different do two stimuli have to be before a person could reliably say that they're different? And by reliably say, we typically mean get the answer right some fixed criterion percentage of time in a forced choice sort of experiment. But in any case, conceptually, that's what a threshold is. And in color, if we start at 3 dimensions, then we could think about, say, a color stimulus that points here. And we could ask, how far do I have to perturb the stimulus to reach threshold in any direction around that point? So basically, what are the set of points around any given reference? What are the set of comparison stimuli that are just noticeably different, that are just at threshold? And we know from a lot of research on measuring these kinds of thresholds in color that a little ellipsoidal blob of color space around each reference contains points that are below threshold, and then right on the surface are points that are equally discriminable, and outside they're above threshold. And so that's this point. Isothreshold discrimination contours, the surface and color space of stimuli that are equally are well approximated by ellipsoids. And there are people who really focus on conditions where perhaps this approximation isn't perfect, but it's a pretty good approximation. We're going to buy into it for this rest of this talk, or at least I am. And this is just saying this shows us an example of a threshold, and people have measured these threshold ellipsoids under various conditions and color, and so we can make those measurements. And they're telling us something about the geometry of color space. They're telling us something. By local, I mean local to a particular point, because they're telling us how far do I have to go in this representation before the stimulus is, by some well-defined criterion, just a little bit different. Now, if we knew the thresholds in every direction around every point in color space and the cone space or some transformation of it, if we knew the thresholds and could read them out, that would really provide a complete characterization of this local geometric structure, this local around every point distance. And really, I think that I would argue that understanding that is sort of the first step toward understanding things like, well, how different are two things that we can easily tell apart? How different do they look in their colors? But we'd like to start with this local thing, and then toward the end of the talk, I'll tell you what we've kind of learned, we think, or how we're approaching this question of whether the local structure does tell us anything about things that are further away. And the challenge, and so people have made measurements of, say, these ellipsoids at a few places in color space. The challenge is that there's a lot of reference stimuli in color space. As shown here, the isothreshold ellipsoids are known experimentally not to have the same shape when you measure them around different reference stimuli, not to have the same size, not to be oriented in the same way. They're very heterogeneous. And so to really understand this local geometry, we've got to understand how that varies throughout color space. And that's a challenge because there are a lot of different references, and there are three dimensions worth of references. There's kind of a cursor dimensionality that starts building up here. You got a lot of references, you have a lot of directions around each reference, and so a lot of thresholds to be measured. And if you think about how long that would take to sort of densely sample all possible references and all possible directions around it by classical psychophysical methods, it will be, I don't know, the heat death of the universe before you're done. It'll be about 1 million trials per subject. That's a lot of trials. And this problem was recognized quite early. Happen to love this quote. This is a quote from Ernst Schrödinger, same Schrödinger of the cat and other kinds of things, but who wrote some very nice thinking about color space and its geometry in the 1920s. And this is an excerpt where he's saying what I just told you. We'd really like to know the local thresholds everywhere in order to build up a theory that goes beyond that. And then he says, unfortunately, this way, by which he means measuring them all, is in its completeness is hopelessly difficult as far as experiment is concerned. And then he turns to try to make up some formula that would describe this based on a sparse number of measurements, which was an enterprise in color vision, actually quite active through starting with Helmholtz, who worked on this, and then Schrödinger, and then Stiles, and Warraven and Voss and McAdam and other people worked on this, but without really the amount of data that you would need to characterize it everywhere. And that's what we decided we would take on as this hopelessly difficult problem. And it was because Alex, as I say, came up to me and said, I've got some efficient methods that maybe we could do this. And we think he's right. I think he's correct. And I'm going to tell you how we extended these current characterizations to give us a complete characterization in individual subjects of these local color geometry. And the idea that we're leveraging is that if I measure the threshold in one place in color space in one direction, if I change the direction a little bit, the threshold's only going to change a little bit. And if I move the reference over a little bit, the ellipsoid's only going to change a little bit. It's all going to vary smoothly. And that, in psychology, it's pretty hard to find super sharp, discrete things. Even a detection psychometric function increases smoothly. So we're going to leverage smoothness. And what that means is that measurements at one point in color space tell us also about what happens nearby. That's essentially what smoothness means, and we'll formalize that. I'll say that other people have had this idea and have done some nice work on it. These Chevalier and Ferrup tried to interpolate McAdams measurements from the 40s. Jan Kunderink and Andrea van Doorn and Carl Gegenfurtner have been making measurements that and using theory that are actually very similar in spirit to what we're doing, different in many details. They have a 2018 paper and a 2026 preprint that you can read if you really get into this and think about how it's the same or different from what we're doing. Okay, so how do we proceed? Well, if we're going to collect data, and the data we're going to collect are going to be forced choice color discrimination trials. We decided we wanted to make the measurements using stimuli that took a step toward, viewing in the natural world. So objects that had surface color. And so what we render these three blobby objects. The screen sits there without any blobby objects, but these three little cubic rooms in it. And in a trial, three objects appear. Two of them are rendered with the same body color, and one of them is different, and the one that's different is in a random location, and the subject's task is to say, where's the different one? One of three buttons. So which one? Yep, bottom right from your point of view. Great, that's an easy one. How about here? Getting harder. Maybe, yeah, we got arrows going in both ways. Actually, it depends a lot on where you stand. There's enough angular dependence. I think it's physically that one, but I can never keep, actually, it looks different on my screen and does over there. And this is a hard trial, near threshold, maybe below threshold. So what do? We bring a subject in, and over a period of time now, we run about 42,000 trials of this sort. And 42,000 is a lot, but it's not a million. And as you'll see, we've got 3 subjects who've now done this for us, and we have a few more who are continuing to do it. It costs us a little bit of money per hour, but not too much. So this cube, you'll be seeing a lot of cubes. This cube is a representation of the display gamut of our monitor. So monitors mix red, green, and blue. The values go from off to on. There's a red channel, a green channel, and a blue channel. And so all the stimuli we can present are contained within this cube. We happen to parameterize it for reasons that shall remain private to me for the talk, from -1 to 1 instead of from 0 to 1, but it's just the monitor gamut. And you can see the trials are spread out within the monitor gamut. Each point is a reference, and then it's connected to a comparison that describes a trial the subject ran, and we know whether they got it right or wrong. We use an adaptive psychophysical trial placement procedure developed at Meta called AE-Psych, which you can download and use if you like, to try to put them in smart places. We also intersperse other trials when AE-Psych times out and doesn't tell us what to do by the time the subject's ready to go. We put something in, and so we collect this data. No 2 trials the same, no repeat trials in this, and that's because we really want to sample the full space, and then we're going to use this smoothness idea to bring it all together. And how's that going to go? So we interpret these data using what we call the Wishart process psychophysical model. I'll tell you just a little bit about the flavor of that, because we would use up the whole hour if I took you through all the steps and the details. It is, I will say, described in what is now, I don't know, anybody who's published in eLife recently has learned that publication is not a punctate event. It happens gradually over time. It's published in a sense, and it's not final yet, but it's a reviewed preprint that you can go read, and the revision is on bioRxiv, and the revision has been basically accepted. Referees said we did great, and so that'll be coming out in its published form. But all the technical detail and quite a bit of data in a kind of warm-up set of this stuff where we just worked within a plane of color space with 2 dimensions to make it a little bit simpler are in that paper, but none of the ideas change when we go to 3. So first thing is this something called a finite base Wishart random process is basically a prior over fields of covariance matrices. So at every point in the monitor cube, we assume that the observer's performance is limited by multivariate random Gaussian noise, an idea that you're probably familiar with from the theory of signal detection in one-dimensional spaces. Internal noise is driving thresholds. In color, that noise has to be 3-dimensional, and we're going to have a covariance matrix, which will be, therefore, an ellipsoidal isoprobability contour, and we're going to try to figure out what noise explains the data best. And so this Wishart random process thing provides priors over any possible field of covariance matrices, and then we choose particular parameters for the prior so they vary smoothly. So that's part one. We developed and generalized for our purposes a kind of multivariate theory of signal detection account for how, if you know the noise for two stimuli, how often will the subject get them correct in our odd one out task. That's pretty standard. We use some numerical methods to compute it in reasonably fast. And so that gives us a prior over the covariance field, which if we knew, we could predict all the thresholds. It gives us a way to say for any choice of covariance field, how often would the observer get any given of our trials correct? And we can sum up, that's the likelihood of an individual trial. We sum up the log likelihood of 42,000 trials. That gives you the log likelihood of the data. And then we turn a big Bayes crank with a lot of computing that the Flatiron Center makes available to us. And that gives us an estimate of the threshold ellipsoids everywhere in the monitor gamut. And I'll say a little more about what those look like in a second. But just to say the same thing I just said, but now with pictures instead of words. So I've said it twice, but still not really explained any of the technical detail. We're Bayesians all the way down. We're going to have a prior where sigma, and this just illustrates a two-dimensional version, and these are meant to be sample isoprobability contours of two-dimensional bivariate Gaussian noise, but in this talk, it's really all three-dimensional. It's just harder to show. You have a prior that says those fields are smooth. That's P of capital sigma. The actual posterior distribution, the distribution we think describes that, given a bunch of data, is the likelihood given any given sigma that we produced our data. That's the theory of signal detection model, which assumes that the subject sees three draws, two of the reference, one of the comparison. They're each perturbed by noise. Then there's a decision rule that says which two are the closest. The one that's furthest is the odd one out, and we simulate out what the probability correct is. That gives us a likelihood for any given trial. And then we multiply that by the prior. That's the posterior. We use numerical methods to estimate the maximum of the posterior, and that gives us an estimate of the covariance fields, to which we then apply this to produce this field here of threshold ellipsoids everywhere in the monitor gamut. I'm just showing it to you on a 5 by 5 by 5 grid, but you could point anywhere in there and say, what's the ellipsoid there? And we'd go read it out and could tell you, because the description is a continuous description of how that noise varies everywhere. So this is the thing that Schrödinger said hopelessly difficult to do, with perhaps one important caveat, which is, well, why should we believe it's actually right? There's a lot of trials, no true trials were repeated. You used a lot of fancy machinery you didn't explain. You showed me this picture and claimed it was right. Is there any reason to think that's true? And we worry a lot about that. And so what we did in this particular experiment is actually, it wasn't just 42,000 trials. Interspersed through the whole experiment were another 6,000 trials that were not used at all in fitting the Wishart model. And what we did was we picked 25 different references shown here. And for each one, we set up a method of constant stimulus measurement of a psychometric function. So we picked, say this one, here's the reference. And then we moved out in a direction, also picked at random for this subject. And there were, I think, 11 different points here where we sort of guessed a priori we thought the action would be in terms of percent correct. One catch trial, really easy. We just use that to monitor that our observers are paying attention, which they do, even after 42,000 trials. And we have then measured with these black points the percent correct. I think it was 20 repeat trials per point, all interspersed throughout the length of this experiment. Every once in a while, one of these trials, well, 6 out of 4,800 times, one of these trials appears. 6 out of 48 times one of these trials appear. And so by the end, we've got 25 psychometric functions. Here's a typical Weibull function running between 33 and 100%. Here's our criterion at 67%. This is our kind of classically no smoothness assumed estimate of threshold from these interspersed validation trials that don't assume any of this Wishart process stuff, and then and its error bar. And here, this purple is our readout of performance along this same line from the Wishart fit, which those ellipsoids were also readouts that we just read those ones out. That's the purple line. It's a little stochastic because of this Monte Carlo way we do it. And the purple region is you can see they really overlap very nicely. And we can look at a summary of that agreement by plotting I'll call it classically measured threshold, validation threshold. We got 25 of those. And the threshold that we read out from our Wishart process. And each of these is one of those points with a 95% confidence interval. We put a regression line through it whose slope is 0.99 for this subject. 20 out of 20 of the confidence intervals overlap. So that was all looking pretty good. And so it's this kind of thing, it's actually exactly this thing that makes us think, since we didn't choose these in any particular, that our threshold ellipses are really a good description of the actual performance everywhere. We've, as I mentioned now, done this with 3 subjects. So we've got 3 sets of ellipsoids. You can see, if you just look at some of the gross features, they're small down here in the center, just sort of toward the 0, 0 point of the monitor. They get bigger going that way. That's kind of Weber's law. I'll show you a slice in a second. And you can see those features are sort of the same. You can see some differences in the observers. This one is longer than that one, and maybe let's see some other things that differ. This ellipse is skinnier than for these 2 subjects. These 2 subjects look similar up here, but maybe a little different over there. This subject overall is more sensitive. So there are some differences between the subjects, but we now have these data sets to, I'll say, enjoy and exploit and use that we think really give us a nice constraint on the local geometry of color space. One thing that we thought, so I'll just tell you a little bit more about the data itself, just some observations from it. In color, there's a concept of isoluminance, which if you're familiar with, you know exactly what it is. And if you're not, think of it, although Steve Chevelle is listening out there, he'll yell at me later, think of it as about the same brightness. But we can take a slice, isoluminant slice in our monitor gamut, read out the ellipses in that slice, and then flatten it out to look at. And so this is a look at, from the 3D data, of what the ellipses look like in what's called the isoluminant plane of color space. And the background plots to the center of this particular representation. So that's where the subject is adapted. And the first thing you see is that the threshold ellipses are smallest near the background. And that, as a general principle, has been observed in sparser measurements of color thresholds since at least, I don't know, for a long time, but let's say since at least the 70s. And they get bigger as you go out in different directions. So that's one feature you see here. Another feature you see is it's just not that they're smallest here. They systematically get larger no matter which way you go. And they sort of are biggest around the outside, the more saturated colors, less precision. People who think about efficient coding might say, well, those are because those you don't see them as often as the ones near the center. We haven't done that analysis in any formal way. They also have the feature that they're all kind of distended, you might say. They're all looking at the center. If you think of each, I don't know, it's a little bit anthropomorphic, but they're all kind of rotated around, so they point to the center. Some people like to call this the superimportance of hue, because as you go around this way, you're changing what we might think of as the hue, and they're a little narrower in the polar direction than they are in the saturation direction. That's a state you have to be a little careful about, because if we allowed ourselves stretching of the color space, it might not remain true in all orientations. But these 3 features of the data, again, people have observed before with smaller, sparser measurements, and so we feel like we get a better look at it here, but it's quite consistent with what's been known before. I'll also point out the colored ones are the slices here, and the little dashed ones are an independent set of measurements with the same subject we made, just measuring ellipses in this plane from the eLife paper. And so the fact that the 42,000 3D data agree with the earlier, again, totally independent trials, same subject in this slice, not perfectly, but any of you ever measured a threshold on Monday and measured it again on Friday, know that they can be a little different. So it's not shocking that they're too different. Give us, again, reassurance that this method is giving us a good characterization. But we do have to keep in mind a little bit that it's not infinitely precise. Just for fun, a question I sometimes get asked, some of you may sometimes get asked, how many different colors can we see? Maybe the popular media would call up and ask that. And these data actually let us give at least a qualified answer to that question. We've got the ellipsoids everywhere. We know we can compute the volume of any one of them. So we just read them out on 125 ellipsoids throughout the monitor gamut. computed the volume of each of them. That turns out to be 0.002, the average volume. Well, this cube, I said I parameterized it from -1 to 1, so its volume is 2 times 2 times 2 is 8. I divide those two numbers. You could stuff 44,000 of these ellipsoids into that, and you'd have equal the volume of the cube. So roughly speaking, if you depend a little bit on your criterion for how much could they overlap before they were a different color, but 4,000-ish perceptually distinct colors visible in the monitor gamut. And there are a lot of caveats with that. Color thresholds depend on how big the stimuli are and how long they're on for and where they are in the visual field, where you're looking. But it isn't a million, it isn't 10, it isn't 100. Sometimes you hear millions of colors, and just to dispense with that, that's a calculation of the number of stimuli, about 16 million distinct physical stimuli you could present on an 8-bit monitor where R, G, and B were each coded as 256 levels. So you get the cube of that is about 16 million. So when you hear millions of colors, it's a physical number, not perceptual. And I will say that our 4,000 number is consistent with an estimate from that 2018 paper of Kunderink, Van Doorn, and Gegenfertners that I mentioned. They were in a couple thousand for different methods, different stimuli. So that's sort of, I don't know, sort of a little interlude here. Let's talk about mechanism a little bit. So we started, I showed you data of this sort, sliced through the isoluminant plane, measured against a gray background. These are actually measurements that we just made in the plane for a new subject who was not in our paper. But it looks just like all those subjects. And then what we did was we changed the background and we put a yellow background on, but measured exactly the same set of stimuli. And what you see, again, consistent with what we think would happen, is that the smallest threshold, here's the yellow background in this. Now the smallest threshold is over here rather than over there. If we go to a blue background that's up here, now the smallest threshold is moving up here. And the other thing you see is that the ellipsoid, it's peaking over at the yellow background here. It rotates a little bit to the gray background. It rotates even more to the blue background. They kind of, this peaking in at the adaptation point and the getting bigger away from it is consistent across these different backgrounds. We have a really nice picture of that now. Now, one thing we know about the encoding of light, I told you about the cones, the cone excitations, is that even in the cones themselves, there's gain control and a kind of conversion of the representation to what we might call contrast. Contrast being the difference of a stimulus from the background divided by the background level computed separately in each of the classes of the cones. So we've got measurements for 3 backgrounds. They're complicated. We don't know what causes the ellipses to do what they do. But if the first thing that happens is a conversion to cone contrast, we can normalize each of those data sets by plotting it in a contrast representation and ask, well, are they the same after that, or do they still vary a lot? And so we've done a simple version of that where what we did was we started with the data. Let's say in this one, we got the monitor coordinates, we computed the cone coordinates, we computed cone contrast with respect to this background, and then to make it a little more familiar, to just keep it looking the same, we then went back the other direction with respect to this background. So we used the contrast ID to map from here to here and from here to here, and we plotted the ellipses on top of each other. And I think this is quite hot off the presses, but what you can see is that contrast coding is bringing the 3 data sets into much better agreement than if we hadn't applied it. So it's getting much of the effect, this adaptation effect, is being accounted for by a mechanism which we did think happened early in the visual pathways. And you can also see that they're not exactly overlaid. There are some differences. They're not always quite oriented the same. And so we'll be spending some time. Here's another example where they're kind of not trying to see what other features of a kind of mechanistic model we might need to bring these data into better alignment with each other across the adapting backgrounds, as well as to understand why these shapes arise the way they do, which we could talk about later, but I don't have anything to say about at the moment, or at least not today. Okay, so those are some features of our data set, the fact that we are able to make these measurements. And I also will just comment point that we think these methods are not terribly specific to color. There are lots of features of performance where you have multivariate, where the rule of combination sort of leads you to something ellipsoidal-like. And we think these efficient methods could be used to provide fuller characterizations of performance in many different aspects of visual science. Okay, but I want to come back to the question I started with, which is actually maybe stop for a second. I think we've got time. Any quick questions, particularly of clarification? We'll have time for the discussion at the end. Yeah.
Sorry, just about the priors for the
Yeah.
model.
Yeah.
So if I've understood this correctly, the priors is a distribution of every possible field of covariance.
That's right. So a Wishart distribution without the word process is a distribution over covariance matrices of some dimension. And a Wishart process is now a distribution over fields of covariance matrices. So it's every possible covariance matrix at every possible point is assigned a probability by the prior. In some cases, for the particular prior we use, that probability is 0, but for smoothly varying ones, there's varying prior, and the smoother it is, the higher the prior. If we have time to talk later, I could tell you how that goes, but it's described in our manuscript. Yeah, and back.
There's just a question about the demographics between the three subjects.
Yeah.
Are they?
Let's see. Honestly, I'm pretty. We're running 6. I actually don't know who they are. I know subject 1, and I don't think she might is one of the authors of this presentation. The other 2 are young. I know that. I believe, but I'm not entirely sure that one is female and one is male. Not a lot of age difference. I think at least one is Asian and at least one is Caucasian. And I'm not sure about the third. I will say in the long term, so running 42,000 trials per subject on a large sample of subjects, particularly if you want to do it on calibrated apparatus in the lab, be very difficult. We think as we learn more and more about how these threshold fields go, we will be able to impose stronger and stronger priors that capture reasonably what we might expect to be the individual variation, or if we develop a parametric model, and then we could greatly reduce the number of trials we needed per subject. So that's something we kind of have in mind as a place we might be able to go to get much wider samples of how these things vary across individuals as a function of all kinds of factors. Okay, so let's come back to super threshold judgments. That is to say, judgments of stimuli that are now further apart than a threshold, which people will have to come up with a way to measure how different they look. And then we want to ask whether we can build a metric, and we're going to look particularly at what's called a Riemannian geodesic metric. I'll explain what I mean by that. Can we use that to account for super threshold judgments? And this is a long-standing hypothesis. It turns out, so Riemann developed, the geometry of curved stuff like sphere, the globe, and curved manifolds. It's used a lot in gravitation. Actually, in Riemann's Habilitation, where he wrote these ideas down, he mentioned color as a possible application for the ideas. Helmholtz picked this up in the late 1800s. Schrödinger was all over it, and a number of color scientists, McAdam, Ujecki, Judd, these guys, Stiles, thought this was an interesting hypothesis without successfully producing an account that accounted for it. There's been some recent papers that have argued that it's not right. I'm not entirely persuaded by those papers, but in any case, we'll call it the geodesic hypothesis. Can we use a Riemannian metric on color space where we've now measured what's called the line element, which are the threshold measurements sort of define that metric and successfully predict superthreshold stuff. So first concept we need is the concept of geodesic distance, and I'll unpack that. So suppose we have a reference stimulus. Again, I'm going to use a 2-dimensional picture here, and I've got ellipses everywhere, and I have some comparison stimulus over here. If at any path that I take between these 2 points, it could be totally wiggly, whatever, I could add up the number of thresholds that I traversed going across it. So that's actually an idea that, anyway, this goes back to Fechner. And I could count them up. And if I did that for all possible paths, of which there are many, and found the one where that adding them up was the shortest, that would be the geodesic distance. And the geodesic distance is sort of a compromise between going in a straight line, which might cause you to cross more threshold ellipses, and going in a curve, depending on where thresholds were, one threshold distance got you the furthest, and those two things trade off. And there are ways, theorems that allow you to actually find the shortest path computationally. So from our data, we can compute the geodesic distance between any 2 points in color space, and we can do it in 3D. And I'm going to show you some videos that just illustrate the same idea again, at least if they play. But we're going to, basically what we're after is this way of computing distance in color space going to help us. So here's the monitor gamut. Here is a threshold ellipsoid at 1 point. I'm going to, I think, successfully play the movie. It worked. It worked a minute ago. It worked when I tested this. It always, okay. How am I going to play the movie? Where is it? Oh, maybe I have to, okay, we'll try a couple things. How do I not? Okay, we're going to play the movie this way. Okay, so we'll just take a slightly smaller view. So this is a pretty random path that smooth, but otherwise, what's happening here is about every nth, I think nth is 2, 3, or 4. Fang who made this video, showed us the ellipsoid at that point. And since this area, we could kind of count these up. There are, this is the geodesic, this is the shortest of all possible paths. We get 10 of them. So the geodesic distance is 10. We found this by using these methods that allow us to find the shortest path. But I think if I go to this one, here's some other path that is a more wandering path. And you'll see that when you go on this one, there are more. So this is a path that isn't the geodesic distance. Happens to be 14. But in each case, we're just adding up the ellipsoids, computing the distance. And we can do one more. This is now the straight line. If I take the straight line in the monitor gamut and do the same adding up, the geodesic one was 10 in terms of number of thresholds, to take a slightly more meandering path to avoid places where crossing the thresholds are really small and you build up more of them. So that's the idea of geodesics in videos. And what we want to do, and I will go back to, I think that was my last video, is now do an experiment that measures superthreshold color difference and then asks, can the geodesics that we predict from our threshold measurements actually account for it? So the experiment, we wanted to keep all the spatial temporal factors as much as we could the same. So in this experiment, we have a reference, and the reference could be at any of 3 locations, but it's queued on each trial. And the other 2 are comparison 1 and comparison 2. And the subject is asked to say which, C1 or C2, is more different from the reference. So any opinions? Point up. Okay, well, yeah, the orange one, most people agree is more different. And here's another example. It's harder, I think. Some people think right, okay. Some people think up. Anyway, this one's harder, but we, again, just like the threshold measurements, we can just repeat this trial after trial after trial. And these are, I'll call, so the threshold data I showed you were, we pre-registered exactly what we were going to do, how we were going to analyze them, all that kind of stuff I regard as sort of archival solid data. These data are preliminary data where we just wanted to try out some things, and we will, eventually redo this experiment probably slightly better. But here's the design. We picked for each what we call a condition, and we're going to measure, I think, 12 conditions. For each condition, we pick a reference somewhere in color space. We pick a comparison 1, some distance, some geodesic distance away, D1, the distance from the reference to 1. And then we pick a direction along which we could vary C2. And so R1 and this direction define a plane in the 3-dimensional space. And so we're now going to just simplify our lives by thinking about things in that plane. And we can compute in the plane the contour that is equally geodesically different from the reference. It's not an ellipse. It's got whatever shape it has, because the ellipses are getting bigger and smaller as we move in different directions. We pick D1, we get this contour. And the prediction of the geodesic model of superthreshold difference, where R and C1 are noticeably different from each other by some amount, is that the 50-50 point in that judgment, as I go from the reference out in any direction, ought to occur, or at least the simplest prediction, ought to occur when I cross this equal geodesic contour. That is to say, when the thing is on this thing, it's the same geodesic distance from R as it is as C1 is from R. So 50-50 point is the prediction, at least of the say that we call models, geodesic model 0. And so we are going to use again a smoothness-based method to actually estimate the 50-50 contour, but we're also going to interleave trials where we measure a method of constant stimuli in one particular C2 direction. So I'm going to show you those data first. So that yes, it was 12 conditions. So for let's look at one of these panels. The reference when this is C2, this is the distance, actually the geodesic distance between C2 and R, and when it's 0, the reference and comparison to are the same stimulus, and the subject never says C2 is more different than C1, because C1 was some distance away that's just held fixed in the condition. And then as we start moving C2 out away, here's these method of constant stimuli, the percentage of time C2 is picked goes up regularly. And that happens for each of these 12 conditions, which were defined by different references, different C1s, different C2 distances, and different distances to C1. They're all individual conditions. But in each case, nice regular psychometric functions. So actually, we didn't actually know before we did this experiment whether the subject would just produce kind of randomness. This subject, at least, highly regular data for these conditions where we were sort of systematically varying things. And we can pick off the 50-50 point. These are bootstrapped confidence regions. So we've got 12 distances to C2, which we will look at in a second, are they the same as the distance to C1? The other thing we did is we used this AE Psych package I mentioned from Meta, whose goal is actually it's a non-parametric threshold estimation method that includes a trial placement algorithm, and it doesn't assume ellipses or ellipsoids. And we let AE Psych place a bunch of trials in this task and asked it to find us the 50-50 contour. And so all the red ones are ones where, so first we'll put a lot of trials right inside this purple boundary. This purple thing here is its estimate of the 50-50 contour. The black thing is the equal geodesic distance prediction. This is the reference. This is C1, and this is the direction in which we measured the C2 threshold. And so the first thing you see is these red points are where the subject said C2 is closer than C1, and green ones are the other way around. And so you can see that this AE Psych software did a good job of putting points, concentrating them near the 50-50 point, because there's a lot of reds inside and more greens outside, and this purple region is its estimate. So for this condition, the estimated 50-50 point is within, in fact, contains the equal geodesic prediction. So that's a success case. Yeah. The other things to note here, here's the 50-50 point measured by that psychometric function for this condition in its confidence interval. So it's overlaying, although in this case just barely, the AE Psych estimate. And this star is C1. When C2 and C1 are the same, you better be at 50% as well. And so that's well contained within this purple region. So that's good. This was for a case where we went 1.85 threshold units away. So not very much bigger than threshold, just a little bit bigger than threshold. And I would say that here, and this point has to work. And then these points don't have to work, but they do. So the geodesic hypothesis is good for this condition. It works. And I think I've got, yeah, here's another one of about that same scale. So reference, green is the estimated psychophysically 50-50 contour. This is the estimated from the psychometric function, looking good. Here's the point that has to be at 50%. Yes, it's within the contour. And the geodesic prediction, I'm going to call it good enough. Actually, let's see, the geodesic contour. There's the light green circle is the the central, the point estimate from AEPsych. So it's maybe a little off here, but if that was the worst problem we had with this model describing the data, we think it was working pretty well. So for small distances, geodesic model, I would say, predicts reasonably. These are the only two conditions we have measured for less than 2 JND. But we went to larger things, and now you start seeing really systematic violations of this idea. So now D1, 3.5 threshold units, 5 threshold units, same exact format I've shown you. Same, the data all make sense. I think the data are really pretty good. The carefully measured thresholds are in the region. But what you see is that as you move away, here's the place it has to work for. And as you get around to the other side, the 50-50 point is in closer in this direction from the geodesic prediction, which is that point. Over here, it's a little, maybe you'd say it's the same kind of thing that's a little bit skewed. And we have all the cases that are, say, 3 and greater, we see violations of this idea of about this magnitude. So although these are preliminary data, I would say I think they're pretty good data. And this geodesic model, I think, is not going to work out so great for large differences. I do think it's useful to know that at 2 JNDs, if you can predict what happens at 2 JNDs, and that's a useful fact, and maybe we haven't systematically gone further. So that's where we are with this. This is, I say, ongoing research. But I'll just tell you a kind of what we're going to do next. A couple things. First, we're going to get more data. We're going to do it more carefully. We'll do it for more subjects. We have to get the 3D ellipsoids in a subject before we can make these predictions, because actually we'd hope we could do it in the isolumminate plane, but when you compute the geodesic distance between 2 points in the isolumminate plane, the geodesics happily leave the plane and come back to it again. So you can't use the 2D data to make these predictions, even if you're studying in a plane. Now, there's our initial model that I'm showing you the predictions for is kind of a model that nobody who believes in perceptual noise, which we bought into big in our basic setup, would say is right, because really what the subject has access to on each trial is a draw from the internal noise now at 3 points. So the model we should be computing, which we actually have implemented but takes a long time to compute, is on every trial, you take a draw from the noise distribution of the 3 stimuli, you compute the 3 geodesic distances for that draw on that trial, and you ask which one was the biggest, and that's the odd one out, which one was furthest. So we can do that. That's a more reasonable model. These violations look large enough to me that I don't think it's gonna work. Possibly a model that says the neural calculation of geodesic systems is itself noisy, and the noise gets bigger as you go further, maybe. And we'll play around with that a bit. And we also need to compute confidence intervals on everything in sight. So because the geodesic distances we compute are based on psychophysical data, which itself has got some noise. So a lot more to do here, but anyway, I'm telling you where we are. Okay, so this is my last slide. Just what have we done today? So we've developed a novel method, pretty novel method, to provide comprehensive characterization of color discrimination throughout the 3D monitor cube, all directions, everywhere, all the time, all at once. Our methods, we think, are applicable to other characterization of domains. And we think the picture you get when you can see the whole thing, and we're still really just beginning to digest, we've really focused on collecting these data, and we are deeply optimistic that we'll learn interesting things as we look at it in more detail. Fang will be presenting at the VSS Applied meeting that happens after the regular meeting, comparisons of these data to the C-Lab metrics, and show ways that they do and don't agree. They do provide an answer to the question, how many colors can we see, with some caveats. They are helping us, and I think will really continue to help us. This is something we plan to turn to in a big way, formulate and test mechanistic models of color vision, many more constraints on it. And finally, in some ways, I think the deepest, hardest, and question we may or may not ever be able to say something sensible about is, to what degree and how can we relate threshold measurements, which define the local geometry of a perceptual space, to superthreshold measurements, which often are what we'd like to know some things about, how well we can do that. But I think we now at least have the data that allow us to make predictions, which were not available previously. So I will stop there, and then we can have some discussion. Okay, Anna?
Thanks very much, David. Do you think the failure of the super threshold model is due to color naming affecting?
Yeah, I was just I don't know if I told you what I was just talking to John this morning. I said was asking about good ways to measure color naming because we're going to make some measurements this summer. And it's exactly to begin to understand where are those color boundaries, and is it the case that it works okay within a color category, but when you jump categories, do you have to add an indicator variable or do something? I think that's a reasonable hypothesis about why it might fail. There just might be, when you judge them to be in different categories, your judgment of difference might either saturate or have an extra, which way it even goes is a little hard to say, but we want to collect those data for our stimuli in our hands and our apparatus for that reason.
Tasks, so you could have verbal interference or, prevent naming or make the presentation quicker or...
Yeah. Well, you don't want to make the presentation quicker, I think, because that in and of itself we know has effects. So all these measurements are for like one set of spatial temporal conditions, but the naming we can definitely do. The verbal interference is a good idea. I think if we start to think the naming mattered, then a good way to test it might be the verbal interference. The idea of measuring another 42,000 trials with verbal interference is a little daunting, but some of this, we have these very eager students who are happy to spend summer in the lab in dark rooms pushing buttons and yeah. Let's see.
I was going to ask you if you had any thoughts on how the geodesic model maps onto neural responses, but your answer to the last question indicates that maybe it's not at that level like it is.
Yeah. I'll answer your question in an oblique way, which is that Alex Williams, who has developed this whole not the geodesic part, but the Wishart part, is busy using these Wishart methods to characterize cortical, the variability in cortical recordings and responses, not the color stimuli yet, which would allow us to maybe relate the psychophysical discrimination field to a neural discrimination field. And then he has started to think about, but I don't think he hasn't gotten far enough to be able to explain to me. He just says, I've been thinking about it. It's exciting how you might get neurons to compute a geodesic distance and just what a neural model of that might be. But I think we're not. You'd have to ask him. Let's see. Yeah.
Thank you. Really, really interesting stuff. I couldn't help think when you were presenting the results, this last set of results, that when you're computing large scale distances, I mean, it's just a lot of effort. You're essentially having to integrate.
Right.
It's a big computation. So do you think that there's a point where people switch over?
So yeah, so that could be they just do something else. There's another aspect of this whole geodesic model framework and the history of color science that I didn't say anything about, and it's the question, and this actually was the approach that the people who worked on these things really were trying to do, is could you take these measurements and then find a stretching and scaling of the space so they all turned into spheres? Because if you could, then the Euclidean distance in that representation is equivalent to the geodesic distance in this representation. And it turns out, and we're learning, again, at least I'm learning about differential geometry, which is at the heart of this. And as I understand it, and if somebody's an expert, you can educate me if I say this wrong, there's a quantity that we can compute and have computed on this, which is the intrinsic curvature implied by these measurements. And if that curvature is 0, then it is guaranteed that there is such a stretching. And if it's non-zero, it's guaranteed that it's not 0. However, that doesn't answer the question, well, how not 0 is it? And I think the way we answer that is to actually just try to stretch them all into spheres and see how close we can come. That's again on our to-do list. If you could do that, then if the visual system representation did or approximated that, then the computation of distances at least becomes a Euclidean thing and probably easier to do. But the notion that either you just give up and say, yeah, it's big, that'd be one kind of model. You do something, metric to some distance, and then you just say, oh, it's too big, and you saturate, or color categories matter. A lot of reasons why this. And there are also other ways to put non-Euclidean geometries on spaces that don't involve the threshold measurements that might lead to different conclusions. I think that the data that we're going to collect on the super threshold stuff has a purpose beyond testing this model, which is it will be a data set where we'll have the thresholds, we'll have super threshold judgments. If the geodesic distance thing doesn't work out, it'll at least give us the data to start thinking about what might work better. Yeah,
For your model with the 42K trials.
Yeah.
How far do you think you can push it down if you have?
yeah. So I'll say two things about, so that really is a question of, so can we develop a, there's a really talented research fellow at the Flatiron Institute who's working with us on trying to do that. And he's taking the approach of finding ways to express priors that look a lot more like our data, but aren't so much like our data that they impose the structure on it. And he also, we're doing what's called the maximum a posteriori estimate. There are ways to get better characterization of the posterior that involve something that has the Langevin sampling is a word that gets thrown around with respect to this that he's trying to implement, maybe even has implemented for this model. So that's one way I think we could, but we don't know how far we're going to get it down. The other thing that we're super excited about, I'll just say this here, is that we think if you wanted to do, which doesn't reduce the number of trials, but it reduces what you can, increases what you can learn for a lot of trials. Like for adaptation, we think we can do impose smoothness on the effect of adaptation by using, say, 1,000 trials instead of 7,000 per plane, but do 10 or 15 planes, and then assume the effect of adaptation itself is smooth and get them all for about the same number of trials that we're getting 3 at the moment. We're not entirely sure how many trials we need for that. So that's actually making the experiment bigger, but increasing what you learn. That could apply to spatial frequency, or temporal frequency, or other kinds of things, eccentricity, other kinds of things. I take time for one more. One more, yeah.
Is there a world in which you essentially produce a group, a generalizable group average of this, rather than have to?
Yeah. Yeah, it's a really good idea. So there's a number of people, and I think the person who comes to mind for me is Zhonghen Liu, who've really been working on what they call hierarchical Bayesian models. So you sort of have some broad prior about the population, and then you have a bunch of individual measurements, and you simultaneously fit the individuals and the overall prior in a way that tries to, as efficiently as possible, characterize the individual variability and the kind of population mean. I don't see why, at least in principle, that kind of idea could be combined with what we're doing. So that's one approach. We're aiming to get about 6 subjects, and they're all going to be young in this initial data set, and then try to pursue some of these efficiency things next tonight. But that's an idea that we also, oh, here it is. We've also been measuring some dichromats, but we'll talk about that.
Thanks very much for coming.